Welcome, everyone. Thank you for joining us for today's webinar, Bio-Rad Unity as a Solution for Molecular Quality Control Data Management. It is presented by Duane Newton, Ph.D., Director of the Clinical Microbiology Laboratory, Associate Director in the Division of Clinical Pathology, and Professor of Pathology at the University of Michigan. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Bio-Rad Laboratories. For more information about our sponsor, visit www.biorad.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located in the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Newton. I will now turn the presentation over to him.
Thank you, Judy, and thanks everyone for joining us today on the seminar. I'm happy to be presenting. So without further ado, I'll get started. I just wanted to disclose to everyone some of the support that I've received, not only from Bio-Rad, but also from a consulting company that works together with Bio-Rad. I'm on the scientific advisory board for Bio-Rad, and they've also provided me and our lab with support for some of the information we're providing today. So this topic is really important to me, and I wanted to share with you a few points of why I wanted to present. So as you all know, QA programs are important to ensure high quality testing for our patients, and we have to track QC data not only to ensure that we're doing the right thing, but there are regulatory requirements from organizations like for our lab, College of American Pathologists, and those organizations require daily monitoring to ensure test accuracy and high quality performance, as well as longitudinal monitoring to look at trends and potentially identify biases. It's difficult to manage molecular QC data. It's very time consuming, and the rules that have been utilized in other areas of lab medicine aren't easily applied to molecular diagnostics. Do you use CT values? Do you use log data, for example? And up until now, there really haven't been any commercially available products. And so the approaches that we've taken, and you'll see examples from my lab, have been very manual, homegrown. It's very labor intensive and requires a lot of effort. And so that makes managing QC data very difficult, challenging, and that's why it's important to try to identify solutions to help us with that process. So for the presentation today, what I would like to do is show you some examples from my lab is how we manage this data and some of the challenges. I'll also review some approaches that can be taken with Unity, the product from Bio-Rad. I'll also finally share some results of a small evaluation we performed with different commercially available external controls and how Unity was used to monitor that data, but also how the controls perform relative to each other. So a little bit about our laboratory. We're a large academic medical center. We have a 24/7 core lab and an offsite main lab, which is where I'm located today. We have about 20 staff, 10 that are dedicated to the molecular section and 10 that rotate from our main microbiology lab and perform molecular testing. We have a variety of qualitative platforms from a bunch of manufacturers. You can see some of those here. We also do viral load testing and testing for sexually transmitted infection. So almost every platform that you can imagine, probably about 10 or 12 different platforms. And just from frame of reference, our volume of sexually transmitted infection testing, CTNG in particular, is about 40,000 tests a year. And our viral load tests, CMV, EBV, HIV, HBV, and HCV, constitute about 20,000 tests a year of our total 100,000 molecular diagnostic tests that we perform. To give you a little bit of an idea of the size and scope of our laboratory. So the first question that we have is, how are we monitoring QC data? How can we monitor QC data? There's lots of data to keep track of. It's very complex, and it can be difficult, challenging to know when something's actually wrong. One of the approaches that can be taken actually comes from our colleagues in clinical chemistry and using Westgard rules. Westgard rules are applied to assays that have multiple quantitative controls, and viral load tests are a good example in the molecular diagnostics area where these rules can be applied. There are rules that are applied to determine the acceptability of a run, but as I mentioned, they've really been used in clinical chemistry assays, quantitative clinical chemistry assays. So applying these rules to molecular is not easy. It's not straightforward. What metrics are we supposed to use? How do we standardize that? This can be really problematic, but I'll show you some examples of what Westgard rules are and then show you how we've applied them in clinical molecular diagnostics. So this chart shows examples of different Westgard rules. And so initially, these charts are called Levy-Jennings charts. And the thick black line in the middle is the mean. That's the expected mean of the control that you're evaluating. The green dotted lines represent one standard deviation around that mean. The yellow dashed lines are 2 standard deviations, and the solid red lines are 3 standard deviations. So in the box in the upper left corner, you have a run with your control that's run, and it gives you a number, in this case, that is greater than 2 standard deviations above the expected mean. That is the 1S. It's violating that 1-2S rule, which is it's a single value that's 2S, 2 standard deviations above the mean. So that would trigger a warning. It's a yellow flag saying there might be something going on. You need to pay attention, but it wouldn't automatically fail your run. The upper right corner is an example of a violation that would fail a run. This is one where that control value was 3 standard deviations or more outside of the mean. That would constitute a failure and would require some sort of intervention depending on what your lab procedures are. Another example of a violation would be in the bottom left, which is a 2-2S rule. That means that you've had 2 consecutive values for that control that test at greater than 2 standard deviations above or outside of the mean. And then the bottom right is 4-1S rule. So 4 in a row that are at least 1 standard deviation outside of the mean. So these may or may not constitute failures of your run, but they certainly would require some sort of intervention and review just to determine what's going on. But these are the types of rules that can be applied to quantitative assays, and these are the types of rules that we do apply in our evaluation of QC data in our laboratory. The approach that we take is for our viral load assays, and in particular, we're going to talk mostly about HIV viral loads from our lab today. But in general, our assays have, for these quantitative tests, negative controls, low positive controls, and high positive controls. We historically had third-party external controls or in-house developed and external controls that were used in our laboratory developed tests as an extra run control. And really it was used to help us, because they were laboratory developed tests, have an extra level of control and help us pass our runs. Wanted to ensure that even if there was one control that was not performing well, the more controls we had, we felt like that were in range, the more confident we were in that particular assay for that particular run. But those external controls were not really used to monitor performance of the tests over time. It was just that particular, for that particular run. In establishing our data, our expected data that we would monitor, for all of these controls, the quantitative values, we would generate 5 to 10 replicates, or use 5 to 10 replicates to generate a mean and standard deviations to then start to put in place the evaluation of the rules that we would use for monitoring that data. And we program Westgard rules into an Excel spreadsheet, which I'll show you in a moment. But it requires manual entry for each run and manual review on a daily basis. One of the points I'd like to make, though, before we move on, is that we discontinued the use of external controls as a cost-saving measure for our viral load assays because we have almost entirely all IVD assays. They're not laboratory developed tests, and the performance of those tests have stabilized over time and didn't require having an external control to ensure that they pass. But because of that, because we don't have external controls, we really don't have a good way to monitor assay variation over time or to make good lot-to-lot comparisons, except for using previous patients' positive and negative samples. So that's a manual effort as well to collect those and evaluate those. So this is a screenshot, a very busy screenshot, which you're not supposed to be able to see all the values. It's just to show you how complex this process has been for us. So we built this Excel spreadsheet. Each line represents a different day and a different run. We manually input the values that were obtained for that run for the negative, low, and high positive controls. We manually input the lot numbers, and we manually determine whether, then the program determines whether, based on the values that we inputted, whether the run passed or whether Westgard rules were triggered. And then if any remedial action had to be taken, there are comments, there's a box for comments to be inputted. And then on the far right, we have a column where there's documentation of review of those results. So you can see there are examples that are highlighted in yellow or red where there were flags that were generated. There's a couple of examples of runs that failed because of values that were outside of acceptable rules. So this next slide is showing a couple of examples of the Levy-Jennings charts that were generated from Excel, showing the data that we were able to monitor over time. And so similar to what I showed you a couple of slides ago, we have a mean with a thick bar. I think it's the red line through the middle. That is a rolling mean that we're generating, and then you can see the values on either side of that. There are some little hash marks that show you 1 and 2 and 3 standard deviations. And then you can also see the red dots or the yellow dots where there's warnings or rejections. So we're reviewing these. It can be cumbersome and not very pretty because the way we're manipulating the spreadsheet, there might be gaps in the cells because of some changes we've made, or we brought in something new or did something different. And so the drop in the line is just a reflection that there are no data points in that spreadsheet. So it can get ugly from an aesthetic standpoint. So we have these spreadsheets for all of our quantitative assays, and all the data input is manual, as I mentioned, and all the review is manual. So the supervisors and our chief technologists are going into the computer and looking at each of these spreadsheets for all of our assays every day. And it is very time consuming and very cumbersome, and sometimes difficult to identify when problems are really arising or that there are trends that we should be paying attention to. And we've been looking for a long time for something that provided a little more automation or a little more standardization, and became aware of Unity as a product actually from our clinical chemistry colleagues. And it's a commercially available product that has a variety of features that can address the challenges that we're facing in the lab. This product has been available for a long time there's a lot of experience in using it. It allows us and any other user to have a one-time entry of the QC data for the particular lot of reagents. The data entry is pretty simple, and then it automatically calculates the values, the means, and tracks the variances. There's also a reduced risk for manual error because there is interfaceability of the program with your automated instruments. That's very prevalent with the chemistry applications, chemistry laboratory applications. And we're just starting the process with Bio-Rad to establish interfaces with our Abbott instrumentation that we perform viral load testing on. Because of the nature of the software, the data review is very streamlined, and I'll show you examples of some of the reports that are generated and used for review. So the actual meat of the discussion for today is really a couple of parts of a study that we performed in our laboratory started last fall. And the first part was really looking at Unity as a data management solution, and had controls from a variety of different manufacturers that were tested in our HIV viral load assay using Abbott M2000. And I just generated a bunch of data testing those controls and entered that data into Unity and tracked it to see how streamlined that process was, how user-friendly it was. The other part of the evaluation was to really look at the performance of the external controls relative to each other. And I talked about our journey with external controls and decision not to use them for cost savings purposes, but I also pointed out limitations as far as our inability now to track in a very robust way potentially lot-to-lot variation or instrument-to-instrument variation of assays. So using external controls in your assays provides you with an opportunity to do that, and we wanted to start that process of looking at how these controls compare to each other. Were they, when they say they test at a certain value, did they really test at that value? So the different controls that were used were, as I mentioned, our assay was an HIV assay from Abbott. So we had the kit controls provided by Abbott, a negative, a low, and a high positive. We also had a product from Bio-Rad that they provided for us, the AmpliCheck 1 product, which has a negative, low, and high positive controls for multiple analytes, including HIV. Acrometrics has a multi-analyte control that is HIV, HCV, and HBV, low and high positive. Sericare, positive control. And then we also used a lab manufactured by us control that was generated by pooling plasma from a variety of patient specimens that included negative, low, and high positives. So all of these were used in the evaluations that I'm going to describe forthcoming. So the first part of the study was looking at all of these controls tested one time per run over 20 different runs. And we used overall for the entire evaluation, one lot of the HIV assay reagents and sequestered part of that so that the last part 2 of the study could be performed 6 months later, which is now. So the part 1A, we had 20 separate runs where the external controls were run one time. We looked at the comparison of the controls to the stated values, between run reproducibility, and comparison of controls to each other. In the part 1B of the study, we performed within run reproducibility. And so we had multiple replicates of each control that were run on one plate. Aliquots of those replicates were sequestered and stored at minus 30 degrees for 6 months to look at the stability of those controls over time. And then that reproducibility experiment was repeated by our lab recently. I don't have data to share on that, unfortunately, but it will be forthcoming. This is an example of, this is a screenshot from how the data entry looks for Unity. And on the first blush, it may appear, well, this is just as complicated as what I showed you before, but actually it's a lot simpler in that there are screens, which I'm not showing you, where we've already inputted the information for each of the controls that were being used. So the lot numbers and the expected values for those sample for the control was inputted at the front end, and then that carries through by the program in the background. So all that we're doing is at each line, you can see line 1 of the spreadsheet is the 5th of September, and my technologist put in the values that were obtained for that run for that level of control. So the box that pops up is specific for that control. This happens to be the Bio-Rad AmpliCheck control, but there's another box for the Acrometrics controls. There's another window for the Sericare controls and so on. So each of those are able to be tracked separately, and you can see at the top of the box, it tells you exactly what lot number it is, what the material is, when it expires, and the rules, the Westgard rules that we've selected to be included. And then we can put in the values, and as soon as you enter the values, those rules are being applied to it, and you can see there are lots of red where there were failures or values out of range. And I showed this only because I wanted to use it as an example to show how the flags are generated, but the data really wasn't that bad. We just made a mistake when we entered the expected ranges. We put the standard deviation range too narrow, and so a lot of the values came out outside of those ranges. When we went back and corrected it, the data looks much better, but I kept it here for illustrative purposes, and you can see how those flags are generated. The box at the bottom shows you what the inputted means and standard deviations were. So our internally designed spreadsheet, while better than our previous process in terms of labor, is still less than ideal, and we spent a lot of time managing QC. We had, as I told you, we had to enter that data and manually track and process it. But with Unity, if a Westgard rule rejection is triggered, a pop-up box actually is visible, and the technologist has to enter an appropriate remedial action. So not only do you see the red number that's generated here, what you aren't seeing is that pop-up box. And based on what your laboratory procedures are, specific action needs to be put in place based on that variation. And basically serves as a hard stop for the technologist. They can't go any further if there's something out of range. You have to indicate what you're going to do as a result of that out of range value. So this is an example of the Levy-Jennings plots for the same data. And the top is the negative control, level 1. The middle is level 2, and the bottom is level 3 control. So the first thing I wanted to point out is how the flags show up on the Levy-Jennings plots. And so the little red triangles that are circled here represent those values that were 3SD out of range. And so these would have been runs rejected at the time of testing. You can also see the yellow squares, which are circled here. These would be the warnings that would require review. And then at the very top, you can see the codified action. The number there represents the code, and right above the circle, I think you might be able to see it says 30 test assay repeated. So at the top of the chart is a summary of all the information, and any codes that were entered by the technologist would also be listed here. And so when that run failed, then it was repeated. That's what that number is indicated. So you can see an initial review of those charts, or the summary review of those charts, what was the intervention that occurred. This also shows you an example of how this particular data, the level 2 performed relative to the mean. So this control, the expected expected values were 0.2 or 2.3 logs, and it was generally performing higher overall than what was provided by the package insert, the mean provided by the package insert. And so this is an example of how you might or you should approach calculating means internally. So the package insert serves as a guide, but you can calculate the means using replicate testing yourself, and you should do that with each control. And that's what we did, actually. And this is a chart showing that same data recalculated using the first 5 tested values to generate the mean, as opposed to relying on the package insert expected value. And when we did that, you can see that it performed much better around the mean. We did the same thing for the high positive control as well, and it performed very, very well. The interesting thing, or the nice thing about Unity, though, is that it allows the user to determine the approach to take when calculating means and standard deviation. So you can use the package insert data, but you can also use a data range over time, or you can use cumulative data like I just showed you, where you're taking cumulative values from the first 5, the first 10, the first 20. So the user has the ability to customize that and based on the approach that they want to take in their laboratory. So this chart is an example of a report that allows the comparison of multiple Levy-Jennings plots at the same time. And while this example is showing different controls, so the top panel is the Sericare positive control, the middle panel are the AmpliCheck controls, and the bottom panel is the Acrometrics control. You can really use a variety of different combinations of things, and where I see this being of most value is in a situation where, like our lab, where we have multiple M2000 instruments where we are running the same assays. And if you have controls that are being run on the same instruments, for example, an external control in each run, the same external control is being run on different instruments, you can track how those controls are performing on each instrument. So you can associate one control with a particular instrument when you set it up, and then generate plots to compare those, the performance of those, and see if there are any trends that maybe instrument 2 is showing some diminishing values over time, and while they're still in range, you're seeing this trend. And unless you had those controls being run on multiple instruments and you had a good way of tracking it, you might not actually recognize that. Another important aspect of the functionality of the system is shown here with this data review report. And this allows you to review data for a particular control over time. So in this example, you're looking at the Bio-Rad control, level 1, level 2, and level 3. And this was exactly the data that I showed you at the beginning that had all the flags. And I just printed out or took a screenshot of that report. You can see the first one was rejected and the third run was rejected. In this, you have documentation here of not only the values that were obtained, the action that had to happen as a result of the rejection. So in this case, the test or assay was repeated. And we have documentation of the technologist as well as the supervisor review. In this case, this was artificial because this was a research experiment. So the same person was entering data, but you have the ability to have the technologist who entered the data be indicated on the right and the supervisor who's reviewing the data indicated on the left, so that if you're being inspected, you have the opportunity to provide information from one source showing how these controls performed, whether it was rejected, the corrective action that was initiated, and that documentation of supervisorial review. We're in our CAP window right now, and so this is going to be very helpful for us to have one place to be able to provide that information. So now I'll transition into the part of the discussion related to how the different controls that were tested actually perform relative to each other. So the Unity data shown in the previous slides is showed examples that compared the values that we achieved to the expected mean, or within control, how that control performed based on what we expected. But we also wanted to see how the controls perform relative to each other, trying to see where the low and high positive controls tested, that is, what values were obtained compared to the means to each other. And so this first graph shows you how the Abbott, Bio-Rad, Sericare, and our in-house laboratory developed controls performed relative to each other. And so the box indicates, the legend box indicates where we expected those values to test, and the lines are showing you where they did test. And overall, all of the low positive controls tested between 100 and 1,000 copies per mil. Abbott's controls with the kit were right at where we expected them. The Bio-Rad low positive control was a little above. It was testing right around 2.5 logs, and it was indicated to be 2.3. Sericare was around the mean, but overall high. And our laboratory developed was around the mean as well, but overall just a little bit on the low side. The high positive controls, Abbott, Bio-Rad, and our in-house lab controls were all testing right on their expected value. They performed very well. The Acrometrics control was consistently below the expected value and about a log lower than the other controls. So it was testing about at a level of 5,000 copies per mil versus the other three that were in the 50 to 100,000 copies per mil. And so while it was labeled as a high positive control, it really tested more as a mid-range control. I'm not really sure what the dip on run number 3 was. That's something that we're trying to figure out. I don't have an answer for that. But I think, again, what this shows is the value of calculating in your own hands what those, how those controls are performing, and not relying exactly on what the package insert claims are. This next, the next two slides are showing the reproducibility experiment. So this was experiment 1B, where we had, this is just between run reproducibility. So taking that data that I just showed you and calculating the percent CVs over time. So we have, for the low positive controls, the expected means and standard deviations, and highlighted for all the percent CVs for the low positives were in the 2 to 6% range. There are some differences as you can see, but from my experience with low positive controls, where I've seen CVs in the 20 to 30% range sometimes, I thought these all perform very, very well, and I don't think there's really any difference between 6% and 2% when you're talking about 100 to 1,000 copies per mil. On the next slide, then, is the high positive control data. Again, these all perform very, very well. Not a lot of difference in variability from between those controls. Oops. So now is experiment 1B, where we were looking at within run reproducibility. So in this experiment, we had multiple replicates of different controls run on the same plate and the same assay at the same time. And so the numbers in parentheses indicate the number of replicates for each control. And again, as you can see highlighted on the right, there are some differences between the products, but overall, both with the low positive controls seen here and with the high positive controls seen here, there isn't any real difference in my mind between those. So as I mentioned, the second part of this experiment was looking at stability over time. And unfortunately, we're calculating the data right now. My research tech performed those experiments this week, and I was not able to have that in time for the presentation. But we'll be evaluating how stable these controls are over 6 months. That's what the packaging shows, and that the expected stability is 6 months, and we want to evaluate that. So just to go back to this concept of external controls, I mentioned it a little bit in the beginning of or in the middle of the presentation, that, are going back and forth between using them and not using them. And really, there's some good reason to use external controls. You can look at changes in assays across reagent lots or instruments. You can identify issues related to any matrix. So if there's some carryover within the extraction process that doesn't remove inhibitors, or there's a problem with a particular specimen type, it's feasible that these external controls would pick up some test performance differences. Also extremely useful to have well-characterized, standardized reagents that are available for validation of FDA-approved assays or verification of a laboratory-developed test. And I think that standardization, that convenience, where you've got a commercial resource to use, really adds a lot of value. And even for laboratory developed tests, first and foremost, having a commercial source for assay controls, low positive and high positive controls, that's really important. And that's been an approach that we've taken with our LDTs in the past as well. So a variety of scenarios where external controls can add value. And we're looking at processes to start to incorporate those more into our quantitative tests. So just to summarize the discussion today, I think you've seen that there are lots of opportunities to utilize more automated tools. I'm not sure the approaches that other labs are taking, but even using a Excel approach like us is still very cumbersome. And so any solution that could take the technologists back to the bench and away from the computer managing data, I'm all for that. It's a much better use of their time being technologists as opposed to administrators. I think Unity, I hope I've shown, is a commercially available multi-feature option that you can use to track and manage QC data in a very streamlined way. And as I mentioned, the automated capabilities when it's interfaced to your instrument enhances that ease of use. On the second half of the presentation, I presented data on different commercially available controls that are out there, at least with the HIV viral load test that I showed you, the data from the HIV viral load test that I showed you. All these controls perform relatively equivalently, but I think just to reinforce that measuring those controls in your own lab, in your own hands, on your own platforms, and determining what the expected value should be is very, very important. External controls can also add value, as I highlighted on the previous slide, to your molecular quality assurance programs. And together, these controls and Unity can streamline the approaches that you take to really functioning at a high level in a quality assurance program in your laboratory. And finally, I'd like to thank a lot of folks that helped. First and foremost, Scott McClellan, who is our research and development tech, who actually ran all the experiments, generated all the data, and shared that with me so that I could present that to you today. I'd really like to thank Beth Lewis from Bio-Rad, who's the really go-to expert on Unity and has helped us tremendously in getting it implemented in our laboratory. I've had finally a lot of great experience working in partnership with Bio-Rad and NaviDx as a consulting organization, and I really appreciate all of their support. And I thank you all for your attention and your time, and I'm really excited to be able to answer your questions when you have them. Thanks.
Thank you, Dr. Newton, for that informative presentation. I would also like to thank Bio-Rad Laboratories for making today's educational webcast possible. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us. We'll see you next time. Goodbye.